Greetings children, welcome back to the biology class. Today we are going to learn the second part of improvement in food production. Already in the previous class in improvement in food production we have learned about green revolution in detail. So I hope you remember what is Sonalika Kalyan Sona, IRRI, about IR8 rice, and Jaya and Ratna varieties of rice. Sonalika and Kalyan Sona are the varieties of wheat, okay, semi-dwarf varieties of wheat and rice. Now in today's class we are going to learn about crop improvement. This is today's topic, crop improvement. Crop improvement, if this is just a basic technique which we could understand by the word itself. Now, for example, like if someone is talking about your improvement or our improvement, what do we think of? The improvement generally has to be from the desirable traits, desirable characteristics has to be improved. For example, if you could dance, you'll be learning dance in depth. You're learning furthermore steps of dance, furthermore forms of dance. Then you are improving yourself in your skills. Right? So in general, if we are talking about improvement, we need to improve the desirable traits or desirable characters. So for a crop improvement, that particular crop's desirable trait has to be improved. That has to be converted into a superior variety of plant. How do we say that that particular character is a superior quality or that particular trait is a desirable trait? That is based on our dependence. So in case if you are talking about rice or paddy, the better quality rice and the one which gives more quantity rice will be considered as a superior quality. Right? The same, the one which could grow in a disease resistant ability, the one which could show disease resistant ability, the one which could show drought resistant ability, because now we are facing water scarcity throughout the world, right? So when a plant can resist water scarcity, that is considered as a superior variety. So this character determination or the trait determination depends on our need or the farmer's need and then the society's need, right? So a plant which will be improved in its basic desirable trait, that is what crop improvement. So crop improvement the plant can't do by itself. It can do, but from wild varieties what the plant is now is actually an improved variety. So we have to understand that this crop improvement, the particular thing what we are talking about, includes artificial manipulation. We have to improve the crop. We means the farmers or the scientist has to improve the crop based on what is our need. So crop improvement, it's actually a basic plant breeding technique. It's not a new technique. Even though we are learning about that, it's not a new technique. This is already existing topic. You have studied about Mendel's hybridization technique. What he has done is plant breeding technique. You have learned about nomads, like when they are before 10,000 years ago, when they started doing these agricultural practices, they chose some wild varieties. At that time, wheat, rice, those things were wild varieties. When they felt like these plants can be utilized, these cereals can be utilized for their survival, they started domesticating it. They started cultivating it. So this cultivation, when they were doing it, the plant by itself, it undergoes some kind of change. It improved itself. And then some of the practices were done by those people too. Right? So that's how the superior variety of crops have been produced. Now, the major steps of plant breeding. What are the steps in major steps in plant breeding? How do we do that? Crop improvement is a kind of plant breeding only. So what are the steps in plant breeding? Is the first one is collection of variability. What this variability means different traits, different characteristics. Okay, so collection of variability means, let me go with an example so that you can understand better. Like for example, I'm going to improve a plant of like a mango plant, right? In mango, you know, there are a lot of varieties of mangoes are there, like Neelam is there, Alfonso is there, and a lot of varieties are there. Okay, maybe I'm not saying all the varieties, but you know that there is a lot more varieties of mango. Now, collection of variability means when I am selecting some plants for hybridization technique or plant breeding technique, I need to go with variable varieties. So, one plant, I'm choosing plant number one or plant A, which is with high yielding variety, which can like one harvesting time in one yield, I can get nearly some tons of mango from one particular mango tree. So that is one variable. So that is the first one, which can produce huge number of huge number of yield. And the second one, the size of this fruit, size of the fruit is like one fruit can give you 1 kg, for example. So that is a best opt one. So that is another variable. And then the quality of the fruit is so good in one, and the quality of fruit is not so good in another one. So the quality of fruit which gives a very good fruit taste is the next variable, right? And the next mango, which is like disease resistant one, no pest or no germs are attacking that particular plant, it's naturally or it's genetically it's disease resisting one. So there is another variable. So collection of variables includes collection of different traits which we basically prefer, right? So there is collection of variability. We need to collect all the variable varieties. And one important thing in this is the variables has to be selected genetically. Maybe sometimes the farmer is using good manure fertilizer, the soil is good manure, or the soil itself is having too much of good fertilizer or manure by itself naturally, biofertilizers are available, then it may produce good kind of quality quantity fruits. But if we change that, it may not. So this is like a phenotypical change. But we should select a variable which is of genotypically changed. From gene level, you have to check that. So how to check that? It may take 1 or 2 generations to check that. Okay, so this is a time consuming process. Plant breeding techniques are not so easy, even though we are saying it is in simple words, but it's a time consuming process. So first one is collection of variability. We need to collect variables, we need to collect different characteristics. So that is first one, collection of variability. And the second one, evaluation and selection of parents, because we are going to do the hybridization technique. That is our prime aim. So what do we need to do is, as I said just before few seconds, because of some environmental characteristics, some of the quality may be enhanced, which can be considered as a phenotypical change. But we need only the genotypic change. So what do we need to do is, we need to evaluate whether it is a genotypic change or whether it is just a superficial change. How to do that? We need to do crossbreeding. We need to do, pure variety has to be raised. Remember the word like pure line selection. Remember the word that is in genetics. In 10th standard, you would have studied about that. Pure breeding has to be done. So a plant has to be crossed by itself. Like self-pollination has to be done for more than 3-4 generation. And then we need to take the seed out of the plant. If it is still continuously producing, still it's able to produce the same quality, then it is pure line selection. So this evaluation and selection of parents. So selection of parents includes the pure line selection. That is, we need to do self-pollination so that the pure breeding variety has been raised, and then we are doing that selection. Okay, so this is to just expel the character which is just superficial. We need to go with the character which is found in the gene. That is the idea behind this selection and evaluation of parents. And then the third technique is hybridization among selected parents. So now we took genetically super quality parents. And then the next technique is hybridization. This we can do by artificial insemination, that is cross pollination. From one plant, the anther has been selected and it would be dumped onto the stigma of another plant and it has to be bagged. Okay, so this is hybridization technique. And then next one, selection and testing, is for the young one or for the next generation one. So from the seed, or it will be producing fruit, that particular plant will be producing fruit or seed, and then this will be further tested for the progeny testing. That is, the progeny will be tested whether the progeny has the desirable quality. Sometimes, as it is this hybridization technique, we are not doing particular gene hybridization. We are doing just superficial hybridization technique, because can you understand that sometimes when you transfer the pollen from one plant to another, the undesirable traits can also be transferred. If you are expecting one plant which is producing bigger size fruit and another one is very tasty fruit, and you are hybridizing these two plant, you may get smaller size with distasteful one, right? There is a chance to get that. So this thing is what included in selection and testing. We are going to select the superior quality of the progeny, and we need to test whether the progeny possess the superior quality or not. And the next one, again testing, release and commercialization. So after testing, again you have to test that to check whether the seed is being produced and that particular thing is able to produce another seed. That particular thing is, again, it's fertile, again it's producing the same quality. It's not by accident. If it is really in the gene, we need to test it again, and then we are releasing it and we are making it as commercialized one. Okay, so these are the major steps in plant breeding, right? So today I have completed just one page. The first one is crop improvement. So you need to learn crop improvement, you need to read that. And the next one is major steps in plant breeding. You can go with the main objectives of plant breeding also, that is given in a separate box. That is, why should we do plant breeding technique? Already I discussed those things. We need higher yield, we need improved quality, we need disease control variety, and we need control development of this resistance variety against cold, frost and salinity. We need to go with a plant which is able to survive in this particular situation in a desirable way, and it has to meet out the basic need of a food. So these are all the main objectives of plant breeding. So I need you to read these 3 paragraphs: crop improvement, major steps in plant breeding, and main objectives of plant breeding. So this is your immediate work today, and your homework today is learn lesson 24. Already I gave you lesson number 23, organisms and their environment, and now you need to go with lesson 24, that is population interaction. Let me give you homework on every day. Okay, I felt like I need to give you homework every day, just 5 questions. You have to learn and write. That's your choice, no problem. You have to learn and write, or else if you see the questions, at least at that time, please refer the questions, answer, learn and then write. Do not copy. That is the only request I'm having. Just learn and write. Okay, so that's your homework. Lesson 24, population interaction, is your homework. Okay kids, I hope you understand the concept what I taught you, and stay at home, learn proper and complete your homework, and meet you by 6:30 and in Google Classroom. So 6:30 to 10 the homework page would be open, so you can complete your homework and message me back. Okay, thank you kids, stay safe at home. Thank you.